how appropriate it is for us to have the pleasure of having Chris Austin talk with us from NCATS today. He's really helped accelerate the development of new therapies, and setting up the platform at NCATS is the first director. It's very exciting to all of us that know him and know what he's done at NIH with trends, with the knockout mouse project, and other initiatives like the molecular lab libraries project. I mean, he's done an incredible job to think out of the box and create a platform at NIH to help us all do our jobs and help accelerate therapeutic development. So it's my pleasure to have an opportunity to introduce Chris. We just funded, the Alpha One Foundation just funded through our Alpha One project, our venture philanthropy platform, a fellow at NCATS who's going to be working right in the lab there, and with a focus, obviously, and an expertise in Alpha One. But Chris is the one that created that program, that opportunity, and I'd strongly recommend that all the organizations in the sector really look at that seriously, and it helps support NCATS and their mission, and it helps us in our quest for new therapies and cures. So it's my pleasure to introduce Chris Austin. Unfortunately, he's in Washington, D.C. Unfortunate for him. We're glad that he's there because he's looking after things, but 12 inches plus of snow and canceled flights, he's not able to be with us today. Jill called an audible, which is why I'm standing in front of the computer screen here today. So if you're going to ask questions afterwards, we definitely want to have time to ask questions. Get to the mic, and Jill will repeat the question so Chris can hear it and respond. So it's all yours, Chris.
Well, thanks so much, and I'm really sorry I can't be out there for all the reasons you heard. I was really looking forward to getting a break from this whatever vortex we're in at the moment. It's an Arctic vortex or an Antarctic vortex or something. Yeah, it was quite the snowstorm we had yesterday, so everything pretty much got shut down. But I was particularly looking forward to being out there because of the leadership that the National Health Council has had in helping to get NCATS started and then helping us find our way. And as you all know, I think Merle is on our advisory council, and I have asked her to take on a role, which I'll just mention briefly, that's very relevant to what I'm going to talk about. So I'm going to run through quite quickly a lot about what NCATS is doing, and I of course don't have time to go into the science, but we'll rather try to focus on the really critical role that patients and patient groups have in everything we do, and how we're trying to increase that further and make it a standard for not only everything NCATS does, but make this, try to promulgate this idea beyond the walls of NCATS. So this is the problem that I always start with. It's the problem that you're all very aware of, and so I don't need to tell you about this, that we live in this paradoxical time where we know more about ourselves and health and disease than we ever have, but on the other hand, Americans as a population particularly are living shorter and less healthy lives than they should and need to. And certainly not all of this, but a great deal of it is due to the fact that we simply don't have a good process. We don't understand the science, and we don't have good operational systems to transition the real wealth of information on the left to patient benefit on the right. And there's all kinds of outcomes that knock-on effects of that. You certainly see this in the difficulties that the drug device diagnostic industry has had over the last 10 years. Since I left Merck, the pharmaceutical industry has laid off over 200,000 people. And that's not because of need, not because of lack of need in the patient community. It's because of the inefficiencies in this process that are making the the development of new drugs and diagnostics and devices just increasingly untenable from a financial standpoint. I think you're all aware that the clinical trial system in this country is a mess, to put it mildly. And importantly, even when when new interventions get demonstrated as being useful, we're actually terrible as a system in getting those interventions to all the patients who could benefit from them. And so the results are sort of in the red there at the bottom. People are unhealthier than they should be. But also funders of the biomedical research enterprise, whether it's public or private, have either become impatient with us or have lost patience completely. And I always say, whenever I talk to folks who are in this business, that I think this is it's a completely solvable problem. We can win that confidence back, but I think there's some things that need to be done to make that happen. This is the other thing that another trend that I worry about. If you're not familiar with this is an interesting paper to look at. It briefly demonstrates the monotonic decrease in efficiency of developing new drugs since 1950. This is, and Eroom's law is Moore's law spelled backwards. You're probably all familiar with what Moore's law is. What's happened with new drug development is the opposite of what's happened with transistors on microprocessors. Perhaps the most frightening thing about this is if you think about the epochal changes that have happened in health and in medicine and science since 1950, and none of them has bent this curve. And so into this maelstrom comes NCATS, and NCATS was formed about 2 years ago now, and this is the mission. I don't need to read this because you can see it, but I just want to emphasize a couple of words. The first is catalyze. We are a catalyst in more ways than one. First of all, we're 1.8% of the NIH budget. So like a good catalyst, we need to be present in small amounts but be very productive and make the other parts of the ecosystem work better. That's what a catalyst does. They bring together reagents in your body from a biochemical point of view that would be inert otherwise, and allow them to produce products that wouldn't be produced otherwise. And so that's really how NCATS thinks, and it leads to one of the fundamental principles, which I'll keep coming back to, which is really relevant for today, which is that everything NCATS does is a collaboration with somebody. We think a lot about who our customers are, and our ultimate customers are the patients, full stop. And that's something I am very insistent about here, and is somewhat different from some of the other organizations you might be familiar with. So we're all about innovative methods and technologies. It's very clear to us that though we have all 7,000 diseases that we're responsible for, we can't possibly address all of them. So like a good catalyst, we have to develop new methods and technologies that will help other people enhance their translational efforts. When I became director, I did make a small but important modification. This had to be informal because changing the mission statement of an NIH Institute turned out to be more of a bureaucratic process than I expected. But it's important that it's not good enough for us, if you go back to the previous slide, to just develop and implement diagnostics and therapeutics. First of all, we need to be able to focus more broadly on interventions, which are including behavioral interventions, for instance. And we can't just develop them. We have to show that they tangibly improve human health, or else our job is not done. Just a couple things about how NCATS fits into the ecosystem. This is actually a slide that was made in the early days of NCATS. I like to view this as, these are the 27 institutes that make up the NIH. I like to view this slide as an aerial view of the silos that make up NIH. And NCATS was envisioned as a horse of a different color, certainly, but also fundamentally a convener, an adapter, an integrator. We think about what is common to diseases and organ systems rather than what is different about them. We're big believers in the connectedness of science and disease, and that if we approach diseases that way, we will be able to make headway and advances much more rapidly. But because of the position that NCATS has within the NIH ecosystem, which is perhaps best explained by the fact that NCATS does no basic science, We're the only institute at NIH that doesn't. We are full-on translational people. That we're a little bit to the right shifted, if you will, in the development spectrum. So we have much more extensive and routine interactions with advocacy groups such as all of you, nonprofits, the FDA, pharma, biotech, VC, et cetera. And one of the things that we think about a lot, which again is one of the reasons that patients are so central in what we do, is that this standard model of starting with the basic research discovery and going to improve human health is both historically inaccurate and scientifically inefficient. That is to say that if you think back about 50 years, it was very difficult to do reductionist basic laboratory work because things that we take for granted, like cloning and PCR and routine protein isolation, et cetera, simply didn't exist. For most of the life of medical research, it's really worked like this, that they're clinical observations, which then move backwards into basic investigation and then lead forward again into patient research, and so that's something that we're trying to reinstitute. Let me try to go forward here. So there's a lot of talk particularly within the CTSA program that NCATS runs, so this term community engagement. So this is a slide that I used when I was talking to the community engagement folks from the CTSA program a few months ago. And the point that I made was similar to the point that was made in the IOM report on the CTSA program that came out about a year ago that Sharon Terry and Alan Leshner headed, which is that from my point of view, there's no such thing as community engagement. There's what we really mean here is communities. And those communities are different depending on the stage of the translational process and what we're trying to accomplish. And so what we do around here is we think very carefully about when we're translating, what are we translating and to whom? You know, the word translation means that you are carrying something over. That's literally what it means. But who is going to receive this data, this product, this improvement in health that we're trying to catalyze the development of? And unless we involve those people in these processes in the beginning, we're probably not going to ask the right questions. We're probably not going to develop the right answers. And even if we did, the people who are the supposed beneficiaries are not going to be invested in this advance, and so therefore may not be interested in actually utilizing it. So it seems quite straightforward to us, therefore, that you have to have a consistent involvement of patients and patient groups from the very beginning. You know, to us, this is pretty obvious, pretty commonsensical, and I know it is to all of you as well. But this is one of the ways I think NCATS is different. Okay, so I just want to mention briefly, because otherwise I'm going to forget it, that about 6 months ago, I established within our advisory council structure 3 standing subcommittees that are in areas that we I and the council thought were particularly important for NCATS over the long term. So these are not working groups; they're subcommittees, they're standing committees. And I asked Margaret and Merle to head this, and they're deeply engaged with us on this with a number of our staff and other people from the council. And what I've asked them to do is to help me and help NCATS with implementing this vision of involving patients in every project that we do. There are others, medical technologies, interactions with biotech, pharma, and VC, which are the other 3. So what are the programs at NCATS? And I'm going to just run through these briefly for you. One is the CTSA program. One is a large program in rare diseases. And the third big bucket is our technology development, tool development programs. A mantra that I have instituted with everything that NCATS does is this one, and so I just want to spend a minute on this. there are many, many, many problems in the translational space, both scientific problems and operational problems. And certainly we are focused on developing solutions to those problems, innovative tools, technologies, paradigms, et cetera. But in our view, it's not good enough to simply develop those technologies or tools or paradigms. We have to demonstrate in individual use cases, individual diseases or targets, what have you, that these novel approaches actually work, because otherwise no one should believe us that these are better than what is the current status quo. And when we do demonstrate them, history also tells us that we can't simply put out a publication or a press release and congratulate ourselves and go on to the next problem and assume that all of the others who could benefit from this new way of doing things will do so without active dissemination. And this is a whole area I know you're all aware of, this whole area of dissemination science, so NCATS is big into this as well. But so you'll see this in a lot of the press releases we do. We start out with what is the general translational problem, what have we done to develop a new approach to this, demonstrate in a disease, for instance, that this is useful, and then what are we doing to disseminate it. This I'm not going to go through, but this just goes through, and you'll have these slides. If you want to think about how we think about patient engagement in the T1 to T4 space, I feel like this is kind of Coles to Newcastle with this group, so I'm not going to dwell on it. Let me just give you some examples of what we're actually doing. So the CTSA program, as most of you probably know, is a very large program. It's the NIH's single biggest program, actually. It's about $450 million a year. It's about 80% of the NCATS budget, and it supports a national consortium of medical research institutions that are supposed to work together to improve the way clinical and translational research is done worldwide. This is a program that we're quite actively evolving, including using the wonderful report from the IOM, which I'll just touch on in a second. This is what the current map looks like. If you want to think about a sort of large trend here, just realize that most of the famous academic medical centers that you've all heard of are part of this program. However, historically, they have worked pretty much separately. There have been a lot of committees and things of trying to tie these together, but they actually have done very little work together. It just wasn't the purpose of the program. It wasn't the way it was run. But NCATS' vision is quite different. That these wonderful places, if tied together into a true national network for translational medicine with a common electronic medical record structure, a common federated RB structure, way to identify patients and key opinion leaders for any disease you might be interested in, have people who focus on adaptive clinical trial designs that are actually not only able to identify and recruit and get patient studies started, but actually use novel approaches to test those interventions. This could be absolutely transformative, and so this is the vision that we're moving forward with. This is the IOM report on the CTSA program that really strengthened our resolve to do what I just said. And there are these 7 recommendations that I'm not going to dwell on. I just want to mention a couple. One is to strengthen leadership by NCATS to really run the program as a program. And the fourth one is to formalize a standardized metric to find out, to measure, is this enormous taxpayer-funded investment actually delivering for the major problems in the translational space that are so acute. And so let me just skip forward to this one. So in order to make that happen, one of the things we've done is to form a working group. Now, this is different from a subcommittee of the sort that Merle is co-chairing. A working group is a temporary group which is commissioned by a council to do something specific. And this is a working group which is working very hard on particularly the deliverables from the IOM from the CTSA program. And you'll notice that the co-chairs that we chose are emblematic of our approach to this kind of work. So you'll notice that Ron Bartek, who probably a lot of you know, is one of the co-chairs. The second co-chair is Nordesis, who's an academic tumor immunologist who runs the CTSA at the University of Washington. And Scott Weir, though he's at Kansas now, he spent most of his life in pharma, so he's really a drug guy. But you'll notice this is really a remarkably distinguished group of people from across the translational spectrum, and they are doing incredible work and will be delivering report to our council in May, and so far they are still on target. We had a meeting just Monday this week, and they're doing amazing work. The thing I just want to point out briefly is one of the things that the IOM report was asked to do was to comment on this term community engagement, and this is taken right out of the IOM report. And their point really was that the CTSA program should do two things. One, to think about communities broadly across the translational spectrum, and secondly, to focus on and to involve those communities in the work from the beginning. But secondly, also to focus on research in this area. And not have what's been done in the past, which you might call sort of retail community engagement, but community engagement to engagement of communities to get specific work done and to show others the way to how to do this. Okay, so stay tuned on that one. So I'm now going to move backwards in the work that NCATS does from the public health back to practitioner-based translation, back to phase 3 clinical trials, and even before that in the preclinical world. And this is the Division of Preclinical Innovation, DPI, at NCATS. And the vision here is that this is a collaborative pipeline where every project is brought to us by an academic or a small company or sometimes a large company or a patient group that is stuck at one or another of these places at the top. Those blue arrows at the top indicate places that investigators or patients come to us and are stuck. And in each of these boxes is a program that has a peer review entrance review criteria done to get into them, and if you get into them, the purpose of the project is not only to deliver a useful tool or data out the bottom. You notice deliverables at the bottom. And those are either data or a physical reagent like a small molecule, a probe, a repurposed drug, a lead, an siRNA, et cetera. But we like every project to tell us something general about how to do the whole process better, because this whole process, as we all know, is just horrendously inefficient. And so we really are very particular about these projects moving the whole field forward. As well as being useful to our collaborators. So I mentioned that all of them are collaborations. This part of the organization actually has 300 collaborations with people over the country and the world. Most of this goes on at a place called the NIH Chemical Genomics Center, which works in the sort of assay to target to lead space on unprecedented diseases and rare neglected diseases. The Alpha 1 project is actually going on in the building that you're looking at there, and we have had incredible success working with patient groups in this context. And I'll just give you a couple of examples of what we like to call patient-driven science. This is a program that we started a number of years ago with the Charcot-Marie-Tooth Foundation. On identifying of drug modulators for the fundamental defect in CMT1A, which is a gene duplication that causes a gene dosage disease. Secondly, one of the first ones that we got involved in was with Josh Sommer. A lot of you probably know runs the Cordova Foundation, and you'll notice he's a co-author here down there. You can see him right between Michael Kelly and me. Identifying repurposed drugs for chordoma therapy. And lastly, This is a project that we did with the Marigold Foundation, which is a myotonic dystrophy foundation, on identifying compounds which can interfere with the molecular defect which causes myotonic dystrophy. And in each of these cases, these projects started when patients walked in the door and said, "We have a problem, can you help?" And frequently we and our folks would say, "Well, you know, we don't know anything about the disease, but we know a lot about how to translate. So if you teach us something about your disease, we'll teach you something about translation, and we'll form a joint, mutually dependent project team, and we'll make this happen together." And it really is a magical formula, which we're very excited about expanding further now that NCATS is on a firm financial footing as of the current budget. Something that we did a number of years ago, which is sort of emblematic of this, is that we put together the first complete non-redundant list of every drug which had ever been approved for human use worldwide. If you want to do repurposing, for instance, across the entire disease spectrum, and if you want to answer the question, which we do, which is if you take the 5,000 or 6,000 or so rare diseases, just as an example, rare diseases that have no treatment, and you ask yourself, how many of those would be treatable by something in the current PDR, current pharmacopoeia? That's a really interesting question. It's a really important question from a public health standpoint. It's a really interesting question from a sort of systems pharmacology standpoint. But the only way to ask that question is to have every drug which has ever been approved for human use. Nobody's ever done this before. And yes, why? And the reason really is this silo-driven thinking that the cancer people had cancer drugs, and the blood people had blood drugs, and the neuro people had neuro drugs. And one of the first examples that we applied this to was actually in a collaboration, again, and you're going to get tired of hearing me use that word, but a collaboration that we set up in a novel structure, which we called the Learning Collaborative, which for reasons you can imagine, between NCATS and an academic organization at the University of Kansas and a patient-driven research organization, the LLS. And the first drug that we focused on here is a drug called Aranaphen, which is an old rheumatoid arthritis drug that, through work at the NIH, at NCATS, at the NCG, at the NIH Chemical Genomics Center, showed that rheumatoid arthritis drug was very effective in treating a form of chronic lymphocytic leukemia. And then it has been repurposed again to a different kind of lymphoma, and then to a different kind of cancer again. So this is a great example of a drug which had not been considered to be a cancer drug, but actually turns out to be a very effective one. The TREND program that a lot of you probably heard of is a program operationally analogous to the NCGC. That is, that the NIH Chemical Genomics Center works with collaborators who have a target or a cellular phenotype or a disease defect, and they want to go through the early stages from assay development, high-throughput screening, chemical informatics, medicinal chemistry, to make a probe drug, a proto-drug. And the TREND program has the same model, same collaborative model, except it picks up where the NCGC stops and goes to first-in-human studies. And I'll just show you briefly the portfolio, and I don't want to go through this in any detail. I just want to make a couple points. First is that the therapeutic areas are extremely broad. If you look down the list on the left, it's everything from hematologic disease to cancer to infectious disease to muscle disease to neurodegenerative disease. About half the collaborators are academics, and half of them are small molecules. But in the majority of cases, there is a patient group who was involved in these, and I'll just give you one example. And that example is this one. This is a rare neurodegenerative disease, Niemann-Pick C disease. These kids die of a rather horrible combination of sort of a combination of spinocerebellar ataxia, Huntington's disease, and Alzheimer's disease by the time they're in their mid-teens. And this is a project that was started when Cindy Parsegan originally, and then Jonathan Jacoby and a number of others who are parents of kids who have NPC came to us, and with their their involvement as a critical part of the project team, we put together this very large collaborative project team across multiple NIH institutes and universities, and worked through identifying and then getting into patients this very unconventional drug, cyclodextrin. And I just want to show you what the project team looks like. That this, whenever I try to get this across to folks, that you know, this is not individual investigator science. You got to have a team to do this. I always show this slide because this is actually not even the entire project team. This is a picture that was taken after we went to a pre-IND meeting at the FDA. The last thing I just want to mention, and again, I don't have time to go through everything that NCATS is doing. There's a lot of other technology things that we're doing, but I did want to mention the ORDR for a couple of reasons. First, that the ORDR has been a leader in involvement of patient groups for a long time. This is due in large part to the vision of its newly retired director, which I'll get to in a second, Steve Kroft. The Rare Diseases Clinical Research Network, or RDCRN, is very different in that it actually requires involvement of patient advocacy groups as a condition of even applying for a grant. And the data are quite clear that this is one of the reasons this program is so amazingly productive, despite having a quite small budget. The research network is focused on development of consortia that focus on groups of rare diseases. This is another thing I think is sort of ahead of its time, thinking not of a disease as diseases as separate entities, but what is common about these diseases. Again, the direct community engagement of patients as research partners is something that's been a case from the beginning. And this is what the current network looks like. And you notice at the top here is the Coalition of Patient Advocacy Groups. It's a broadly collaborative organization, not only with advocacy groups, but also with all of the other NIHICs in red in there in the top left. And you'll notice that the way these consortia were put together is not on a single disease, but on a group of diseases that are characterized by pathology of a particular organ system, or biochemical pathway, or cell type, or a type of structure in the body. The Genetic and Rare Disease Research Information Center is another thing that is a jewel of this program. It currently gets about 500 inquiries a month from patients, and mainly patients, their families, and healthcare providers, many of whom have just gotten a rare disease diagnosis. And finally, the rare disease registry and repository is something that we're very excited about expanding. We're doing currently a lot of study of this pilot program over the last few years to see how this particular pilot worked in this particular contract. But the point I want to leave you with is that the issue of patient registries and repositories using common data elements and structured ontologies is, we think, absolutely critical to making headway, and to make it possible for not to have 6,000 or 7,000 different registries of different diseases which don't talk to each other, but rather have an integrated system that will allow us to identify commonalities among diseases. So I couldn't give this talk without pointing out that last Saturday, Steve Kroft officially retired. I think probably everybody in the room that I'm talking to knows Steve. He has been an icon, a visionary, an incredible leader of this enterprise since before the Orphan Drug Act was passed over 30 years ago. Has been at the FDA, at the NIH, at the HHS, at the White House, and I'm glad to say that that though he is officially retiring, he has agreed to spend about half time with us, so he's not going away completely. And as he retires, I've been able to give a lot of wonderful valedictory addresses for him and with him. And though I know that one of the ones that he's the most proud of is this one that he got from NORD at their anniversary. I think a lot of you were at last year. But it's important that ORDR, though Steve is moving on, it's going to continue its important work. And my new deputy director, Pema McGinnis, is the acting director of ORDR while we search for a replacement. Its founder is riding into the sunset. Those of you who don't know why Steve is riding a zebra, find someone with a rare disease, and that person will tell you why he's riding a zebra. But again, he's not going away completely. He's going to be back with us, but he has told us that his wife has a honey-do list, which is going to keep him busy for about a month. But after that, he should be able to get back with us. So I'm going to stop there, and I wanted you to have the program lead at NCATS for all these programs that I've mentioned, and in addition, programs which I haven't had time to talk about. I'm serious when I say that they are all collaborative programs, and like with the Alpha 1 project that you heard about at the beginning, many of these projects start from contact with folks like you in the room, so we're always anxious to hear from you. And if you haven't heard enough about NCATS, you can go to our website, follow us on Facebook, you can follow our tweets and other things. And I'd be glad to take any questions if we have time for that.